Brilliant. Well, I'll get started with the introduction then. Hi, everybody. Thank you so much for taking the time to join us. My name is Rachel. I am the Communications and Impact Manager here at UCL's Social Biobehavioral Research Group, and I'm delighted to be supporting the GRACE-Epi programme, including this webinar series. And I'm just going to give a quick introduction before handing over to our speaker for today. The GRACE-Epi webinar series is all about sharing different methods, and it is happening on a monthly basis. And this is part of our new research programme, which is the Global Research on Arts and Cultural Engagement, Advancing Epidemiological Methods, or GRACE-Epi for short. And this is a major new 7-year programme of work all about arts and health, funded through a Wellcome Discovery Award. And if you came to our first webinar in the Monthly Methods series last month, you would have heard more about this from Professor Daisy Fancourt. And there's actually a recording of that webinar available on our website, or you can learn more about the programme on our website. So there's lots more information there. And throughout this programme of work, we are engaging in open and collaborative science. And as part of this, we're hosting these monthly free training webinars, which are open to all. And these are considering novel methodologies, theoretical approaches, and lenses run in partnership with different universities globally. We'll be recording these webinars and then sharing them as a resource for people to watch back via our website. So if you'd like to be notified about future monthly methods webinars, then please do subscribe to our newsletter. We'll be announcing the March one very shortly and sharing that with you so you can sign up. On that note, we are open to suggestions of future topics to cover in the Monthly Methods series. So this is an opportunity to have your work spotlighted as part of a global research programme and connect with different colleagues across countries and disciplines. So it could be that you are wanting to share quantitative, qualitative research methods from any different discipline, but they could be transferable to the field of arts and health and have a relevance to epidemiological or broader public health research. And we have some suggestions of what you might outline in the webinar. To find out more about being a speaker for a future session, then do get in touch with me over email. I put it on the slide here, or you can, I'll post it in the chat towards the end of the session as well. But we definitely love some ideas as we're planning the rest of the schedule for 2026. Super. So on to today's session then. We will shortly be hearing from Professor Agustín Ibáñez, who will be giving a presentation on the topic of a multi-aging clocks framework to study arts, culture, and brain health. And this presentation will be followed by a Q&A. So do please use the Q&A function to ask any questions, and we'll also endeavour to pick up any comments in the chat as well. So by way of a brief introduction, Agustín is one of our investigators for the GRACE-Epi research programme and a global leader in brain health. He serves as director of the Global Research Networks at the Global Brain Health Institute, Trinity College Dublin, scientific director of the Latin American Brain Health Institute, BrainLat, Universidad Adolfo Ibáñez, and his research bridges computational neuroscience, aging clocks, exposome science, whole body health, and artificial intelligence to advance understanding of brain health across diverse populations. So he's authored over 500 publications and is the recipient of major international grants, NIH, NIA, Wellcome Trust, Alzheimer's Association, and he leads multi-center initiatives such as REDLAT and ClipBrain, promoting equitable transdisciplinary approaches to precision brain health worldwide. So we're really lucky to have him with us as a speaker today. So without further ado, I will stop sharing my screen and hand over to you, Agustín. I think you're just on mute.
Apologize. Thank you so much, Rachel, for your kind introduction and for the opportunity to share these ideas and methods to link brain health with arts and cultural engagement, in my case through aging clocks. So I will share my screen. Can you see it, right?
Perfect.
Wonderful. So just a second. Apologize. So those are my disclosures, and what I will bring for discussions with all of you today is first try to bring the link between art and health from a specific point that I want to discuss, that is how we can really measure the impact of arts and cultural engagement, why it's so difficult, and what should be the best framework for that. Then I will focus in the Epi GRACE project and in one specific approach that we are going to develop, like that is basically using different aging clocks from different aspects, from body, brain, behavior, to link to find associations with specific cultural activities, artistic activities. And then I will try to set the future agenda, what we expect to get with that. As you know, Daisy and Nisha Sagnani and others have been done recently, there is a increasing attention of linking the arts as a way, to improve health, to create social prescriptions, to understand arts and cultural engagement as a proper health behavior, right? And I think that we are facing a unique moment. The new Lancet series will start soon, trying to bring in this overall framework, which has been really, really difficult to develop for two main reasons. For one side, I would say biomedical sciences have a strong bias, I would say, against arts, because sometimes they are not considered measurable, or because they are not standard or classical prescriptions things. But the other side, prejudice from the artistic side, has also important role in this, in the sense that for many years, many artists have been questioning the use of arts or cultural engagement as, a way to understand health, or ascribing unmeasurable things to this. So beyond this prejudice, I think that there is a unique opportunity nowadays for using arts. I just want to show this picture because it's from San Juan, the place where I am, is my birthplace. And this is an example as we use puppetry to teach more than 5,000 children across rural schools about, how to develop brain health prevention and protecting their minds. But beyond this small piece of history, what I want to bring is, first of all, why beyond these two, let's say, big prejudice that I mentioned, why it has been so hard to connect arts with health, right? And probably one of the main reasons is that there are not linear associations between artistic activity. First of all, artistic activities will have an impact depending on the meaning, on the purpose, on the impact at, inner levels, subjective level and intersubjective levels. And this is not easy to measure, first of all. Second, because there are many pathways associated with arts that cannot be, measured in a unique way or a unified approach across subjects. But third, because our methods for understanding brain health are very poor. And one important thing I want to bring before moving into arts, I want to bring the negative photo of the arts. That is like the cumulative burden, the stress, the environmental demands, the disease. There is a model that is called allostatic interoception that is really important for understand the potential mechanisms for arts. So what's say allostatic interoceptive overload? Basically, the overall idea is that, our body and our brains are constantly responding to environmental demands. And for that, we have multiple biological, physiopathological pathways, like the immune system, that can reduce the immune energy to respond to environmental demands, or cardiometabolic energy. If you have a threat in the environment, immediately you need to be ready to run or to fight. So your cardio and muscular system will change. The inflammatory is another way to respond to a stress that increase the inflammation responses across your body. And the stress, right? The stress that happens in real life is also happening across multiple biological pathways. So our brain is constantly anticipating and responding to environmental demands. And this is a natural process. But sometimes with the stress become, too strong, sustained in time, our organisms start to develop, atypical responses, abnormal responses that are associated with cardiometabolic health, with cancer, with dementia, and many other things. So everyday behavior, in the sense that the everyday context can induce higher stress or reduce the stress, or larger or better or worse adaptations. And this process is not just, let's say, cultural or psychological or social, it's very biological. But it's also very specific, because it's not about the brain, it's about the body. It's about the cardiovascular, metabolic, inflammatory, stress hormone that have very synergetic, interactive effects in the body and in the brain. This is why it's not easy to capture. I have been working in these approaches to try to connect the huge heterogeneity, not only of the measure of the brain, but across the body and in specific environmental demands. So we need to do an exercise of return the body and return the settings, the situaedness to the brain, because for many years we have been just neglecting that. And for understanding the brain, we need the body and we need the environment. I have been working in computational models with great computational scientists like Gustavo Deco and Morten Kriechenbach. And we are starting to develop better metrics to integrate body and environment. But once again, why is that? Because, the way that the body responds to the environment and the ways that the environment induce cumulative stress is having a huge impact in adaptation. And creativity, adaptability, coping with stress, social interaction, that are things that, as we will see later, are related to artistic activities, are really important, because these factors, the way that we manage stress, increase our predictive coding of what's going to happen, and increase, the kind of threats that we can anticipate, or the kind of adequate or inadequate responses that we are going to bring into the specific settings. And this is really important, not only for disease, but also for resilience, as we will see. Now, my older daughter has been living in Colombia, which is a country that represents a unique opportunity to understand this cumulative interaction between environmental factors. It's a country with a lot of narco trafficking, structural inequalities, radicalization, poverty. But this is happening in every part of the world. And I think that conflict is also a negative way to understand what arts do, because conflict increase the allostatic overload, the cumulative burden, and increase the abnormal responses from the organisms, and can be like a negative of what we can try to reduce through arts or cultural engagement. So for that, it's not only, disease or conflict or vulnerability, it's also resilience. But, for a long time, we have think resilience as a pure genetics. We have, multi-omics pathways that induce resilience. We have genes that protects you against to having dementia. But also, you have cognitive, psychological, emotional process that can help to be resilient, in this case, to respond with less stress to environmental demands. But of course, we also have families and communities. The way that one individual manage the cumulative stress through their social connections, family and communities, can represent an extended buffer for resilience. This is exactly the point that we can come to a more methodological aspect. So one way to measure resilience is to understand which environmental, social, individual, and lifestyle process, not only biological, are associated with regarding resilience, but also how we can measure resilience. And one important thing that I have been developing the last 5 years is to develop a multi-organ framework, trying to understand how your brain, your cognition, your overall biological system becomes more younger or older than you are, because it's a direct measure of health. You have your chronological aging, but your lungs can be older or younger than your chronological age. The same for your brain, the same for many other things, and you can measure that. And this is perhaps the most important thing. When we want to think in how to measure the impact of arts and cultural engagement, especially in aging, being able to measure, individuals that have delay aging, that have a younger age than the chronological age, is a direct measure of health that is interesting to capture. For that, it's not about one single brain area, it's not about one single, let's say, physiopathological pathway like inflammation or immune system or reward system in the brain. It's a kind of holistic synergetic process. This is the kind of approaches that the so-called neurosyndemics or allostatic interoception frameworks can try to understand, that they are not linear unidirectional causal links, but really synergetic compound effects that can measure at the subtle change in aging clocks parameters. This also calls for a less reductionistic neuroscience. We cannot understand creativity just looking what brain areas are, activated. That is the most typical way to measure creativity in the brain. We need to understand the process, and we need to understand the huge heterogeneity in the process of the brain. And we need to understand that changes in the brain not necessarily means, more healthy or more vulnerable. So we need to understand the complexity of the phenomena, and we need to integrate that. We need to integrate the brain with everyday body and cultural activities. So it's a challenge, but also it's a wonderful framework to understanding how brain works in real life. And GRACE-Epi is a wonderful project led by Daisy that wants to answer some of these questions, right? So it's a project that have multiple goals or aims. One of them is to understand what means arts or cultural engagement in completely different socioeconomics and cultural settings, like what that means in Africa or in UK or in Argentina, right? So there are multiple factors that we don't understand yet regarding what is art, what is cultural engagement, and what are the processes associated with that. Another important thing is to developing more large data set epidemiological approaches to try to understand the impact of cultural settings and art across, large amount of data that can bring robust results. Then, of course, we want to connect this with policymakers. If we want to do social prescription of arts, right, or we want to develop, preventable approaches to prevent, let's say, dementia or accelerate aging through arts, we need to better understand how to make the translation into policies. So this project is all about this. But there is one aspect that I will work with Andrew Steptoe on that is using clocks for understanding the impact of art and cultural engagement. So what are clocks? The clocks, the biological clocks, are different metrics from different organs. Let's say we can have an estimated cardiovascular clock. How we do that? So I will provide a really, neuroanatomy of how the clocks work later on. But first, let's have a very intuitive way. Let's say that you take a lot of measure of your heart and you have 100,000 of subjects, so you can see how these cardiac parameters are associated with younger or older person. And you can train machines, artificial intelligence machines, to create a normative approach to identify what is the expected age for your heart and what is the gap with the chronological age. You can do the same for your immune system, your hepatic system, your renal, musculoskeletal, pulmonary, metabolic, brain, cognitive, biobehavioral, natural speech analysis. You can estimate clocks with almost everything. The interesting thing, or what we can learn from this, is two things. So first of all, this is a challenge. Why? Because it means that there is not one single clock to understand aging. Aging is a very complex process and depends on the interaction of multiple organ clocks. So there is nothing universal as aging, and this is why probably scientists don't understand what is really aging, because it's a really complex phenomena. This is one thing, but the other thing is that you can use these different organ clocks to predict, for instance, the health, the estimated biological aging. With organ clocks, you can estimate, the mortality. You can estimate the onset of disease. You can estimate, using 3 or 4 organ clocks from the body, you are able to predict not only the age of the brain, but most important, if one individual will develop some neuropsychiatric disorders. So this is the simple approach. And as I mentioned, I have been working in this idea to linking different organs from different parts of the body or different cognitive or social process, and using this as a marker of how vulnerable or resilient is an individual, considering multiple individual and macro-social factors. I will go into it because if you understand this part, you will see what is the approach that we want to develop. So the simple idea is, let's say that some individuals will be more resilient or more vulnerable. That means that some individuals will have younger age than expected, so they are resilient, or they will get older than expected age, so they will be more vulnerable. And for measuring this, there are multiple ways, not a single universal way to measuring aging. So how does this work? So first of all, you need to develop a model that can predict your age. And you can use as an input almost everything. Let's say you can use the natural speech analysis, or you can use pulmonary or lungs information or brain information. For this, with this information, you train a machine. Could be a simple machine, a regressor, or could be a complex machine like a deep learning, and you try to predict the age of an individual. For that, you need a large amount of data, because otherwise your clocks, cannot be really, really well trained. But this is the first thing, being able to predict, to estimate the age. Once that you have been done this, you can improve your model. As I mentioned, I will present later multiple examples of how to measure clocks. And as I say before, you can just do a regression, like taking a list of inputs. This can be, let's say, different epigenetic metrics, and trying to predict the age, right? So you use a lot of predictor to predict a single outcome, that is the age. And for that, there are different procedures depending on when you have large amount of data and very heterogeneous population, you need complex methods like deep learning. I will show them. But if you have less variables and less heterogeneity, you just can use a classic machine learning classifier or even a linear regressor. The challenge is there is no run-through, right? So what means that a person is really older or younger in biological terms? Of course, we always compare the estimated age that the computational clocks bring for an individual against the real chronological age. So this is our more or less ground truth, but it's very fragile. We may need something more. The other thing is heterogeneity. The errors in any classifier are large. So the models are able to more or less predict well the age at the group level or with a degree of probability, but with multiple range of errors in months or in years. The other thing is that they are biased depending where in the lifespan you predict the age. So the younger people tends to have younger than chronological ages, while older people tend to have older than chronological age, and you need to correct for this age bias. And the most complex or challenging is that you can have multiple different clocks, and they can, in some cases, showing different delay or accelerated aging. So this is the overall framework for estimating clocks. There are multiple strategies for dealing with this, but what we want to do with that, what we want to do is simple. is estimating the gap between the multi-organ clocks and the chronological age to understand if that individual is vulnerable or resilient or have better health or worse health. And using the gap, this difference between the estimated and the chronological age to understand what drives this gap. Is this gap driven by, let's say, genetics or by exposome, environmental factors like pollution or climate change, or is this driven by healthy lifestyle? So the people that have more exercise or do more arts or are more creative have less accelerated, younger aging. And then for really developing global settings, we need to understand this variability in the gap across multiple exotypes and phenotypes. Phenotypes, let's say, how this happen in female versus male, how happen in younger versus older, how this happen in people that come from the global north or the global south, and people with high or low socioeconomic status, in people that have better whole body health or worse whole body health. But what we really want, right, is to use in this framework to understand how the artistic skills or behavior, as well as cultural engagement activities, can modulate or not, the biological aging. That's the simple approach that we want to develop. And I will show you what we have been done. So for instance, you can estimate the aging of your brain. And this can be done with very complex things because your brain, you can estimate the brain activity that is very complex, high dimensional. So in this case, we use deep learning of high order interactions that are properties of the interactive, multi-level interactive part of the brain for estimating the brain aging. And we used a large global data set. And we were able to predict the age using brain information, these high order interactions in a very, with high accuracy. This accuracy was good, but then of course they are still individuals that look younger or older. What we found that modulates that. So first of all, if you live in the global south, you tend to have a more accelerated brain aging. So your brain's looking older than your chronological age. If you are female, in comparison with male, you tend to have, older brains. If you have a disease like dementia or cognitive impairment, of course, you will have sometimes many years as, accelerated brain aging. But if you are female, you have dementia and you live in the global south, you can have 10 to 20 years of brain acceleration. So that was a simple first step that we shown. Then we wanted to measure the pace of aging across global settings. So we, instead to looking at brain clocks, we developed something that we call it biobehavioral clocks. The biobehavioral clocks are used multiple information like protective factors, your cognitive level, your functional ability to move in the world, your well-being, your education, or your physical activities. For one side, protective factor, but also risk factors like, if you have cardiometabolic issues like heart disease, hypertension, if you have sensory impairment like hearing or visual impairment, if you have, sleep problems, if you have unhealthy habits like alcohol consumption. So with these protective and risk factors at individual level, we estimated the biobehavioral age gap. What we found in global settings. So African countries have the most accelerated aging, followed by Latin America, then by Asia, then by Mediterranean Europe, and North Europeans have the largest, the more healthy aging, right? And then we wanted to study what was associated with that. And that was the important thing. So we find that things like, pollution accelerates aging, or socioeconomic inequality or poverty accelerates aging. But also we found that sociopolitical instability. When you live in a country that is continuously, experiencing political stress, of course, your allostatic overload will increase. I hope that you can hear because a lot of people around here apologize. In this paper that is going to be published in Nature Medicine soon, we started to understand how multiple environmental factors like physical exposome, like air pollution, green space, temperature, precipitations, quality of water and soil, etc., as well as social factors like, the GDP, the education, the energy demand, the democratic level of a country, the level of inbound migration, how these factors accelerate brain aging across 34 countries. And that we found, we were able, of course, be able to predict the age again with the brain activity, but that we found that different exposomes, different environmental factors induce accelerated aging. But the most surprising thing was that the combination of factors, let's say when you have multiple physical and social exposome, the degree of brain acceleration is exponential. So you can have a huge burst of accelerated brain aging when your environments are toxic in the sense of, pollutants, but also in the sense of social toxins. So this is very important because these effects are associated with larger accelerated brain aging than those observed by having a dementia. When you have a dementia, you can have 5 to 10 to 15 more accelerated brain aging. But when you have a cumulative exposome, you can have an even larger effect. Now we are starting to understand how this accelerated aging in the brain is associated to fundamental mechanisms of brain organization, like, very basic mechanisms like the coupling between excitation and inhibition in the brain. That is a very broad mechanism associated with aging, and it's really, sensible to subtle things. We also shown that genetics can accelerate or delay brain aging. For example, the Down syndrome is nowadays considered a kind of Alzheimer's genetic presentation. We have shown that the people with Down syndrome can have 20, sometimes 30 years of accelerated brain aging, that the degree of accelerated brain aging is directly associated with excitatory inhibitory activity in the brain, especially in areas where multiple genes associated with dementia are expressed. So we also have been shown that the larger the neuropsychiatric symptoms of a person, the larger the accelerated aging, especially in the allostatic interoceptive networks of the brain, supporting the idea that this is a cumulative burden. We have been shown, for example, that yes, the natural speech analysis, with a few minutes of natural speech analysis, we can get 700 features, and with these features, we try to predict the age. We estimate the gaps with the chronological age, and that we have shown is that the speech age gaps can predict very well, the risk of dementia in terms of biomarkers of dementia, your level of cognitive and clinical performance, the impact of social exposome of that, but also with just the natural speech, we can find associations with the age of your brain, with the age of your epigenetic clocks that are biological systemic metrics of the aging of your body. And coming close to the idea of how we can use that is that we don't want to just measure disease. For instance, we want to measure how we can delay brain aging. So the people that use multiple angles tend to have less accelerated aging. The people that do exercise in this randomized control trial, we've shown that the people that do resistant exercise tend to reduce the pace of brain aging. Then more recently, we did this job that was very, very popular because we've shown that people that performs creative activities like tango dancers, musicians, visual artists, gamers, they tend to have, less accelerated brain aging. And even, we tested the interventions, and short interventions can really delay the brain age. So I think that this is the core that we want to develop, showing how specific artistic activities can induce delay aging. So this effect that we shown was consistent across different artistic activities. So it was not a specific activity per se, but was the degree of creativity. The larger the expertise in creative activities, or the larger of the time devoted to activities, was also associated with the pace of brain aging. And then we did biophysical modeling, and we found that the main hubs associated with this delay, younger brains, were associated with really critical process relevant for creativity. So why multi-aging clocks might matter for art? So first of all, arts and cultural engagement are whole system health behaviors. It's not something that is going to modify one brain circuit, one neurotransmitter, one cardiovascular aspect. It's a way, not always working, by the way, but it's a way to, developing ways to reduce the stress to environmental demands, and will have a very systemic health impact. So we need to understand plasticity not in local or reductionist terms. We need to understand how arts can reduce allostasis, this overload biological effort by continuous response to stress, and how brain, body, and environment dynamics occur. Not single brain region, not single pathway. So multi-aging clocks can help for that, because can quantify the impact of arts and cultural engagement in delaying biological aging across different measures. Can capture organ, system, whole body, biobehavioral age, or even brain age gaps, right? So, and can integrate phenotypic, molecular, and exposomic data. So I think that this is what we want to do with the GRACE-Epi project. As you know, they will harmonize large data sets in a very, very, well-represented data. At the very least, 12 data sets across different countries will have biological data in which we will be able to estimate different aging process. Like, we will get epigenomics, metabolomics, proteomics, phenomics, and I hope that we can also develop biobehavioral age gap, and in a subsamples, we may be able to develop brain age gaps. With this, we will be able to connect what we know about these thousands of individuals regarding their own engagement in artistic activities, and how this is associated with delay aging, biological aging, and how this is modulated by different factors like overall health, or exposomic factors like socioeconomic inequality, or the level of pollution where the people lives. So this is going to be a very fun project. I'm looking forward for the next years for doing that. This project just is starting now. They will take about, 2-3 years probably to get this harmonized global data set before we can test the hypothesis, but we are ready for that, and I hope that you enjoy the conversation, and let me know if you have any questions. I think that we have time for conversations, right, Rachel?
Yes, thank you so much, Agustín. That was a brilliant presentation, and I think I'm really fascinated by this approach to looking at arts and health. So thank you. We have just under 20 minutes where we can go through some questions. We already have some that people have submitted in the Q&A, and I can see one in the chat as well, but do people continue to write, and we'll try and get through them. So I can ask the first question from Rin, which is, how are you exploring measurement of salience within the brain related to culturally specific arts practices to determine specific country-aligned, culturally responsive arts engagement?
Okay, I'm not sure if I understand well the question. I think that the question is how to model the effect of cultural and country level difference in this effect of art and health, right? So the quick answer is by having a lot of data and being able to model that. How? So you will have thousands of participants from different countries, in which we will estimate different clocks, biological clocks, epigenetic clocks, biobehavioral clocks. So each individual will have some of this estimated aging, right? Then the first thing that we are going to see is how specific cultural engaged activity that are being collected in the human flourishing studies or in other data sets, how these factors are associated with delay or accelerated brain aging. If we found what we expect, let's say that there is a delay, so the people looks younger than the chronological age when they have, a stronger engagement with cultural or artistic activity, we check the first point, right? So we are able to show that there is an association between cultural and artistic activities and rejuvenation of different multi-clocks. But then, of course, there is the problem of the country. So first of all, we will look at the variability of the countries. I am 100% sure that we are going to have huge effect about country levels. So for these country levels, we can leverage different social and cultural aspect. Like, we can leverage like the overall health, the overall conditions, what's the level of inequality. So the people that live in inequality countries, in more inequality countries, tend to have more accelerated aging. We already know that. So we will see how, if this association of art is more accentuated or diminished in countries with larger or smaller inequality in relation to the effect in the aging process. We are going to do exactly the same with multiple social exposome, the level of education, the level of community engagement. If, let's say, your neighbor is risky, is dangerous, like if you live in Argentina or Colombia, or is completely safe if you live in Donnybrook in Dublin. So we are able to tackling that. But we hope with this data set, we can understand the interactions. It could be multiple vulnerabilities and resilience associated factors in the exposome or country level information that can have, non-linear association, increasing or decreasing the effect. What I expect? Well, in general, I expect that if you live in, underserved settings, you tend to have more accelerated aging, you have a more cumulative effect of the allostasis, so the artistic effect probably will have a smaller effect. I could be wrong, but this is what I expect, because it's not enough to have a kind of behavioral change to make a big change in your biological aging if you have so much cumulative burden. But this is just an hypothesis. We need to test it. Others can think quite the opposite. For instance, in Latin America or in Africa, there is a lot of what we call familismo, like, huge impact of the families for protect individuals, to reduce the stress, to create a sense, a meaning in life, to help them to cope with the stress. So maybe that's effects in Africa or Latin America, maybe the effect of arts in reducing accelerated aging can be accentuated. That is an alternative hypothesis that could happen. So the good news is we don't know, but the good news is that we are able to answer with this data set, right?
Fantastic, thank you. And a question now from Carolina saying, what public policies could help with mental health care in a society like Colombia's that you mentioned?
Oh, that's a fantastic question. So many things. First of all, social prescription is a luxury from a few countries nowadays, especially in the north. Social prescriptions can really help societies. It's very well known. I think that the person was mentioned something about Colombia. Is that right?
Yeah, okay. As an example.
So Colombia have many examples of, Colombia is facing a terrible civil war, much worse than happened in Ireland, for example, with 7 million of victims, 350,000 deaths, a lot of displacements, a lot of, dehumanization process. And arts can really help. So a lot of people use painting or use, narratives or use cultural support to cope with that, right? The trick is, I think that is clear. The thing is how to measure that. We really need to measure the biological imprinting of that, because otherwise we cannot convince policymakers that they should be a change. If the policymakers understand that can reduce, the burden of disease and can save an incredible large amount of money, probably they will put more efforts in developing arts and cultural engagement interventions. So we need to provide evidence to convince policymakers. We need to also establish what type of interventions are better for a specific context that we don't really know. We just have example of these methods in this country or this in this, but we don't have a systematic science of arts and cultural engagement. So we need to push that agenda to really develop a more tailored phenomena. But this is not just for art, it's in general. You know, if you want to predict dementia, for example, the field where I work is exactly the same. We are not able to predict dementia in an universal way. We need to understand, you know, the whole body disease of each individual, the history of the access to the health system, the impact of multiple exposome variables in the overall cumulative burden. So we really need to develop tailored information. So for convincing policymakers, we need to do more local research using local data and addressing the local situated factors if we want to be precise, if we want to produce good science and good markers for intervention.
Super, thank you. And I think that actually links to the next question that I have here, which is saying the research is brilliant, especially the distinction between country and factors that are generalizing brain aging. What initiatives are taking place to translate and transfer the evidence broadly, understanding that this webinar is part of ongoing efforts to do just that? So I know as part of the GRACE-Epi program, we'll be building towards doing policy work and evidence sharing, but is there anything you want to say about your kind of work in influencing policy and practice?
Well, I think that there are multiple examples. Epi Grace is a wonderful one because it will have a full work stream for translation, for translating this evidence, working with toolkits, with policymakers, developing tailored recommendations, right? But there are many others. So Nisha Sagnani at the Jamil's lab is doing so many interventions and coordination with government and social prescription at national level with the US. Be tuning with the forthcoming Lancet series for arts and health that the two papers should come hopefully in a few months ahead, and we'll systematize and summarize that. There is a WHO report on the impact of intervention that Daisy have led in the past that I suggest you to have a look. There are multiple initiatives like the Creative Brain Week that happen in, I don't know, 10 countries nowadays, and so on. In the Global South, there is a lack of systematic assessment. There is a lot of arts and cultural engagement, but this happen in a, you know, wild, uncontrolled way, without measurement, without link to the policymakers. And I think that there is a huge cultural reserve in the Global South that can be exploited for, you know, improving health and to creating policy implications and recommendations. But we are very far away of that.
Thank you. Yeah. So I think watch this space and sign up for newsletter updates and from all of the investigators and research groups involved, and hopefully we'll have more to share. Great. And then next I can see a really helpful comment from Marie Celine, who is commenting on the use of global north, global south, and making the point that different indigenous peoples might face different experiences and could live in both northern and southern hemispheres. So I think that is a really important point to consider. And actually in our first work stream for GRACE-Epi, we are doing a number of workshops which are aiming to kind of interrogate some of our concepts and language and framing. So this is a really helpful feedback that in this first year and throughout the project, we're definitely open to. So thank you for sharing that, and do drop us an email if you want to find out more about the workshops where we'll be discussing things. I don't know if you have anything you want to add to that, or
regarding that point, yes, I think that many distinctions that we like, you know, like separation, what is art, what is not artistic, what is creative, what is not creative, what is rational. Many of these distinctions are very Occidental and doesn't happen in other cultures. And there are a lot of myths about, you know, these kind of distinctions in the brain or in the biological process, the same way that we make a division between arts, you know, doesn't have any, let's say, measurable impact on health. From if you ask people about 30 years ago, most of them will think that in the biomedical field at the very least, right? So this is why it's so important to understand different cultural perspective, because we are following distinctions that could be totally wrong or could apply in specific settings. So understanding how we can incorporate. The other thing is, artists for artists or artists for genius, for Da Vinci's. While in fact, you can use different things associated to arts or creativity to reduce your stress and to have a better, well-being and to have a better health. So this is also important, but also it's important to don't develop oversimplistic approaches, right? It's not because you are going to do mindfulness that you eliminate the risk to have cancer, for example. Of course, you can reduce the inflammation, you can reduce a lot of biological process if the artistic activity is really meaningful. It really helps you to reduce the stress, but you can also have genetic factors that in any case are going to, accelerate a process that you can develop a disease. So this is the other part. We need to understand the holistic interactions. Arts and creativity are one part of the healthy behaviors. Exercise, for example, is another one, right? Or social engagement or social interactions are other parts that are important for having health. And we cannot expect that one single behavior is going to have, the same impact in all ways. And at the same time, when we talk about art and about creativity, some people think that the arts have unique components, that nothing have it in other fields. I don't think so. I think that it's still a discussion, right? Anyone can have their own opinions, but I think that some components of creativity, for instance, reduction of stress, can be, present in other lifestyles, or exercise. When you dance, you can do it also by doing proper exercise, or just stopping the overthinking, or reducing the stress. This can happen by different processes. I think that arts and creativity have the power to combine many different of these salutogenic process that can really help your health, but can be expressed in many different and cultural ways. This is the other thing that we really need to understand, because otherwise we will look at a prototypical expression of art while there are many others that are possible.
Thank you very much. I fear we might have more questions than we have time, but I'll keep going for a bit more. So our next question is from Juan. Was there any specific system or biological clock that was more relevant at the time of generating greater global aging?
No, it's $1 million question. A few months ago, no, I think that 6 months ago or something like that, there is a very nice discussion in the journal Nature. They invited, the world leaders on aging, and they asked, what is aging? And they were not simple agreement. So for some people, it's something that happened, at the cell levels, other things that depends on physiological resilience, other things, completely different frameworks. So nobody knows what is aging, in the sense that we can describe hundreds or thousands of processes associated with aging, but doesn't mean that there is a unified conceptualization. In the same line, nowadays, at the very least, there is not clear pathways of which clocks will be the best to capture the cultural and artistic engagement effects. I tend to think, because this overall effect, arts have this kind of multi-domain effect. If you look at common pathways like inflammation, it's being modulated by stress, by exercise, by your predictive coding. So I think that the inflammatory clocks should be like a general metric of arts and health engagement. It's an hypothesis. We don't know yet. I mean, we have not been done systematic comparisons. And then you will have different artistic activities, like dance, for instance, impacting, the pathways associated to exercise, and et cetera, et cetera, et cetera. But I don't know I could say, I don't know, and it should be a shorter answer, sorry.
That's okay. That's okay. That's very helpful. I think we only have a couple of minutes left. So although I can see there is some, and maybe you can also see we have some questions we haven't been able to answer today, and some people also just saying thank you already in the chat. But hopefully through our kind of subsequent research dissemination and sharing of findings, we might be able to get back to people on some of the topics and questions raised as we're generating more research in this field. Any final words from you, Augustine?
No, I think that I mean, for me, the only thing I will say is I really love this field, not only because we need to develop something more than pharmacological interventions. We really need to develop multi-component interventions. In my field, in dementia, multi-component interventions are the most successful nowadays to prevent dementia, right? And the arts have this kind of flavor in the sense that can integrate different salutogenic process. So this is very important. But for me, it's really important to understand in real life, to understand if we want to use neuroscience, if we want to use biology to understand how we can improve the lives of other people. This is the kind of project that really challenge the current model of the brain, that are isolated model looking just at what happened inside the brain and ignoring what happening at the individual level, at the body level, and at the context or cultural levels.
Brilliant. Yeah, and I think that's the exciting part of this project is it's how so interdisciplinary and global, and looking forward to seeing what happens next. So thank you so much for taking the time, Augustine, for your presentation, and for everybody who has joined today's sessions and asked questions and engaged. I'll be following up over email with some useful links and the webinar recording in the coming days. Do sign up to our newsletter for details of future webinars that you can come to, and we'd love your suggestions as well for future event topics. So I've put in the chat as well, just for now, how you can get in touch about that and a few useful links. But lots of people saying thank you, and thanks to everyone again. I hope you have a lovely rest of your day and really appreciate you taking the time to join.
Thank you. Thank you so much, Rachel. Thank you all for your question and attention. Bye.